Okay, so an overview of the genetic algorithms case study. First thing I did in class is I showed you how to get to the new resources. So /cs small cs 2021, not 2022.php. You'll have to type that in yourself. And then I went through what that gives you. It gives you the case study on a digital platform, but including what on the paper case study are just terms that are italicized. Here you've got now hoverovers, which give definitions of each of them. And each of those words appear at the end of the case study, and I've got the same copy and pasted definitions there. A couple other little miscellaneous things that are extra here, including a video on natural selection, which you should all definitely take a look at. And I think there are a couple other kind of random videos thrown in that aren't hoverovers but actual links. But mainly the hoverovers. But also links to each of the four challenges with their own notes. Okay, so that'll be my part of the bargain with you guys, is to go over each of those to a certain degree, and you can research beyond that. Yeah, and just what else is here is stuff that's not on the case study. So here we go, more case study resources, including sample questions, which you'll definitely do, videos, which you've already seen, IntelliJ project, which is great, you can run it, it's cool, and a bunch of links from other teachers, other students around the world that I think are all right. But I think everything you need is, well, it's up to you, right? You're the researcher to learn all this. So, and then there's a paper 3, information from the IB, may be helpful to you. Particularly helpful, though, will be just looking at past papers, which are only 4 questions, including that last one, that 4th one, which is always worth 12. And yeah, so there you go. So what I will do today, what I've already done today, and I'm just going to do it for the people that missed the class, people want to review, is I think this will work this way. I'll just a bird's eye view of initialize, evaluating, selecting, crossing over, possibly putting in mutations, and getting the best result from your genetic algorithm. So we start with saying that there are these towns that Latte or Fennon want to go to, and the question is, what's the shortest route? Is it this? Might be, but it might not be. So, right, no brute force. We're going to initialize a bunch of populations. Let's say 100. And these are lettered. So one of the random 20 town tours could be what I've got here: B, C, G, E, F, blah, 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 blah. And there are actually 20 of them. So, or another one could be A, D, E, whatever. And another one could be H, whatever. So we'll do 100 of these. That's initialization. While true gets us into the loop. We're going to evaluate. And evaluation in this case, pretty easy, because we just add up all the distances using that matrix. And let's just say that this one was 2,500, and this was 3,600, and this was 2,900, whatever. Okay, so, but we evaluate them. And then usually we're going to rank. And usually it's going to be least to greatest, and so maybe there ends up being one that's 2,200, and one that's 2,300, and one that's 2,450. We don't know what the order is. You just imagine that those are ranked. So having ranked our initial population, then we want to check to see if we've reached our termination condition, which could be, and I'm not going to write this down, this was in class, but it could be that we want to just run this a certain amount of time, or it could be that we want to run a certain number of iterations of this, or it could be that we just want to do this until we get to a result that we're happy with. So let's say it's that one. So let's say that our termination condition, well, I'll write that here. Termination condition is, I don't know, like 1,700 kilometers. So we'll run this genetic algorithm as many times as it takes until we get 1,700. Okay, but we don't at this point, because the best up here was the 2,200. So we keep on going, right? No termination condition. So we select. So what we're going to do is we're going to take. It's probably going to be. Well, there are 100. So we could take maybe the top 50. More on this later, but let's just say that we take the top 50. Take the top 50. Take the top 50, and we cross them over each other. In other words, they mate, as it were, to give the next generation. So if you got 50, you got 50 factorial different combinations of mating or crossing over that can happen. So A can cross over with, this can cross over with, let me get the pointer here. This could cross over with this, or it could cross over with that, or these two could cross over, or we get all sorts of crossover possibilities. And so we just do a certain number of crossovers. I don't know how to undo that back. Too bad. Shoot. But we cross over. We cross over a bunch of them, and we end up with a new population, the second generation, and it's going to, just by definition, be a little bit better. And so we're going to end up with B to whatever. We're going to end up with C to whatever. K, whatever. But this second population, or this second generation, not the initial one, but the second one, I don't know what I just did there, it's going to be better. It's going to be slightly better because we've taken the top 50, right, and we've combined them. We didn't take the roots that were the longest, we took the roots that were the shortest, and we combined them, and we were just naturally, when we do that, going to come up with better roots. And we're going to end up with numbers like 2150 and 2175 and 3200. And so, same idea, we're going to rank these by shortest to longest. We're going to have another, 50 down, or 100 down here, so we're going to have 100 here that were crossed over from the first generation, and then we're going to have 100 ranked, and then we're going to take the top 50, and again, they're going to come up and be the third generation, and so on, and so on, and so on. And I guess the only other main thing that I went over in class, just happened to get there, so I'll do that with this video, is that what will happen quite often is you end up with a suboptimal kind of spinning of wheels, where you get them all being about the same amount, not in the second generation, but maybe in like the 50th generation, still doing the same thing, and you end up being stuck down around, let's say 1700 is our aim. Let's say that we get stuck around 1900 or 1800. Let's say, so that, like, am I even going to rank these? No, I won't rank them. So 1900. And 1950 and 1889, and they're all going to be in around there. And you can rank them, but they're all basically the same. We're kind of stuck. And like I said, I got there. So I guess that the reason that I got there is we need to talk about mutate. So the crossing over is helping. Oh, and I also talked about how you cross over. Just very quickly, in terms of selecting, that's easy because you want to select the best, but you get the best by combining them, and I didn't really show that in the video. And all of this was going to come up in the next month and a half, or well, certainly the next 4 classes. But crossing over, if you had B, C, F, K, M, P, O, etc., and you had another A, J, it can't be K, but we'll talk about that another time, L, D. I don't think we're up to S, but it could be Q, and another letter is not there. So, E. So we're talking about crossing over, how genes, the one gene and the other gene, you take them and you cross them over. And that's what happens with the evolution of life, that the genes cross over. And you end up with the new gene that's part. So we'll just kind of, how do we do this? We'll just circle this. So you get two new genes, and one of them is right here, blue then black, and the other one is black then blue, right? Because they crossed over. So if I were to draw that completely, and maybe when I'll slow this down, do it a little bit better another time, you end up with two genes, but you're actually like black, and that's blue. So see if you're following this. Black, and then I'll do this blue. Blue, then the other gene is blue, then black. Okay, so this is the crossover process, and what we end up with, it's supposed to erase. What we end up with is a mixture, right? And what's going to be nice is if, you know, like this part of blue was really good and this part of black was really good. So you put them together and you have two really good ones next to each other. So let's do that one crossover. So the one crossover to the generation is going to be, let's say we keep the B, the C, and the F, and the K, but then we cross over and we get the D, and the S, and the Q, and the E. Okay, that's crossover. I didn't mention that. That's how you mix them up, right? You're taking the generation up here, all 100 of them, you're ranking them, which I've erased, and then you take the top 50, and you know, one pair at a time, cross them over, so you get new combinations. And so ultimately, if you're still following me, you get these new combinations, but they're stuck. And this is where I got into actually some of the notes, so I might as well finish this off. Talk about mutations, and I think I used convergence. Because what happens is you converge to a mediocre if you're not careful. And I went over how this, you take two pretty goods, what do you end up with? Pretty goods. And you just kind of get to a point where they're all pretty good, but they're not really, really good. And so more on this another time, but you're going to want to throw in not just mutations, which I'll end with, but you also want to throw in randomness of taking not necessarily very good from that ranking of 100, not just taking the top 50, but taking the top 50 plus a scattering of the bottom 50 as well. And you may end up with a not very good, you know, half of it actually being quite good, and then the complementary of another not very good one is really good as well, and you end up with something unexpected by throwing in this randomness, not just taking the best, and you end up with something really, really good. And then finally, even that may not be enough, because you are, and this is a completely different example, but it's an easy way to show how mutations, sometimes the pretty good, even if it's using randomness like this one, you're still not quite to the absolute optimum, because you're not throwing in absolutely every combination. And so one way to throw in absolutely every combination is to throw in not too much, because it slows things down too much, but throw in mutations regularly. And what this is showing is that if you were trying to spell unicorn by combining Unilever, Hello World, and popcorn, then eventually over here you'd be able to, because we took the first three of Unilever, UNI, and the last four of popcorn, CORN, you'd end up with unicorn. But over here, this population runs into a rut where it just can't get over fixing that last letter, because there is no, let's say we had 100 different combinations here, and you can only swap you know, up and down this way, then there is no RN, no, we want to end with an N. We want to end with it. There is no N. So you throw in a randomness of an N and other random letters throughout. You know, the vast majority of mutations aren't going to help anything, but every once in a while, like, ooh, that's exactly what we needed at the end, and now we have unicorn. So all that stuff is what we did in class, just to get a general bird's eye view of genetic algorithms. And in the next 4 classes, we'll go through the 4 challenges and put it all into context.